The
Mr. Duras, I'm subscribed to your YouTube page.
You don't?
I am.
Oh, good. All right, so our next section here is about transport. It's about the movement of molecules, the movement of substances, both within the cell itself and from cell to cell. We will talk more about transport later on as we start talking about body systems and the circulatory system and so on. But for now, we're sort of just talking about the basics of cellular transport. So we are going to review osmosis and diffusion. But we'll talk about some more of the details about how things get around in the cell and get into and out of the cell. So when we look here, we have obviously what structures us? Cell membrane, the plasma membrane. And you can see here that phospholipid bilayer with proteins that are integrated within it. And we'll talk in more detail about that membrane and what some of these proteins do, what their function is. All right, but we'll start sort of with some basics. So first off, just at the molecular level, we have the process which I know you're familiar with, diffusion. And so diffusion is the movement of molecules. And so we're going to go into a little bit more detail here than you did in 7th grade. Because in 7th grade, it said it's the movement of molecules from high to low concentration. But we've added something saying the net movement of molecules. Does anybody know what that adds? Saying the net movement of material. Hello? Yes? Yep. Okay. Especially they have your lunch in 10 minutes. So net movement.
Like how they...
Not so much how, but if you remember when we talk about diffusion, we say molecules go from a higher to lower concentration. Well, actually, molecules are going both ways. Okay? Molecules are always sort of moving randomly with random motion because molecules are moving. And so there's some molecules are going from low to high, some are going from high to low, but overall, the net, the total movement after you take everything into account is from a higher area of a higher concentration to an area of lower concentration. And again, this happens randomly. It does not require any energy. It's just due to the random movement of molecules that happens. Okay? And it continues generally until an equilibrium has been reached. So molecules will go from a higher concentration to a lower concentration. Some will be moving each way, but they will then eventually reach an equilibrium when molecules are overall equally distributed, where you have areas of equal concentration. And even when you have areas of equal concentration, there's still movement back and forth, but the overall movement keeps those concentrations the same. And we have lots of examples of diffusion in living things, and here's a couple examples. When we talk about our lungs, in the alveoli of our lungs, we inhale air that is high in oxygen, and the capillaries surrounding these alveoli, they're lower in oxygen. And so the net movement of oxygen is into those blood cells. It goes from a higher concentration in the air we breathe in into the lower concentration in our red blood cells, and the net movement is into the red blood cell. And at the same time, okay, carbon dioxide does the opposite. There's a higher concentration in the blood, lower concentration in the air of the alveoli, and so the net movement is the opposite. And that is diffusion.
If you were in a room that was completely surrounded by walls so no air could escape, would you eventually die from breathing CO2?
Yeah, I mean, I guess eventually if it's completely sealed up, you could. All right, so diffusion, the rate of diffusion, how quickly molecules move, is dependent on many factors. The size of the particles is one factor. Smaller particles diffuse more quickly. The temperature. Higher temperature results in faster diffusion. Why? Sophia?
Because particles move faster and higher temperature.
Yeah, higher temperature means the molecules are moving faster, which means they randomly bump into each other more and will diffuse more quickly. Pressure. Okay, so the pressure, higher pressure can increase the rate of diffusion. Solubility. Okay? More soluble the materials are, the faster they will diffuse. And the concentration gradient.
Mr. Darrell said only made one hole.
What do you think that means? Anyone want to take a guess as to what concentration gradient means? The concentration gradient. Brandon?
Like how much of a molecule there is in a certain area from one area to another.
Yeah, pretty much. It's the Grace, what were you gonna say?
Like if there's a larger difference between the two concentrations of molecules, then they'll diffuse from the higher to lower quicker.
Yeah, the bigger the difference. If you have a very high concentration of a molecule and a very low concentration, diffusion will take place more quickly. If they're closer, then diffusion will take place more slowly. So the concentration gradient is another factor.
I think it's broken.
That's all right.
There's like a chunk of material like sticking I don't think it likes
So
Wait, Mr.
the other, yes.
What's solubility?
The solubility is a factor of how, yes, oh, this is not this.
maybe the paper.
Of how much of a substance will dissolve in a solvent is solubility. There's also times when a molecule, even though there's differences in concentration between outside of the cell and inside, where molecules still cannot move. They cannot get through that cell membrane. And oftentimes there are certain proteins in the cell membrane that allow those molecules through. So it has to do with matching receptor molecules that can allow some of these molecules to diffuse into the cell. Molecules also diffuse through gases. And diffusion, by definition, doesn't require a membrane. If I place a drop of dye in water, it will diffuse through the water. It doesn't have to pass through a membrane. Osmosis is a special case, sort of a subtype of diffusion. Does anybody remember the definition that we used for osmosis back in 7th grade? Nate?
No, it's just the diffusion of water through a membrane.
Yeah, diffusion of water through a semi-permeable membrane. Like other molecules, water will diffuse from high to low concentration. When it goes through a membrane, we call that osmosis. So here we have two liquids. One has some salt in it. One has more salt in it. Okay? And so here we see these little Mickey Mouse shapes are water molecules. Okay, and here there's a higher concentration of water molecules. Here there's a lower concentration, so water will travel through that membrane with a net movement to the right in this case. Always from higher to lower concentration.
To work?
Yeah. Who has that?
So you know, for some need it.
No, it's not working normally now. I'm getting sick.
All right.
Whoa.
So we talk about solutions and solvents and solutes, and I think you remember what those things are. So a solution, when we say we have a solution, we're talking about a solute dissolved in a solvent. And the molecules are evenly distributed. Typically they are clear, not cloudy, but they may be colored, but they're not generally cloudy. The solute is what disappears. It's a substance that is basically broken down into individual molecules so that you can no longer see it. But it is not a chemical change, it's just a physical mixing of those substances. Okay? Examples of salt dissolved in water or sugar dissolved in water, oxygen diffused in the air, oxygen dissolved in water, nutrients. The solvent is the material that holds the solute. And often we're talking about water. Water is often known as the universal solvent because many, many different materials can dissolve in water. And we have some terms that we use when we're describing solutions. And this takes some thought. Terms we use are hypotonic, hypertonic, and isotonic. Do you know what the prefix hypo means? Like if somebody's hypoglycemic, or if you need to get a vaccine, they may use a hypodermic needle. Sebastian, you know what it means? Where does what is hypodermic needle? What does derm come from?
Germ.
No? Skin. Like dermatologist, skin. What might hypo mean then?
I'm gonna guess like fast.
No?
I thought it was
Cold? I would guess low because like hypothermia.
Yeah, hypothermia. Yeah, it means basically under. Okay? A hypodermic needle goes under your skin.
Oh.
Okay? Hypothermia is when your body temperature drops. Hypoglycemic means your blood sugar drops. So when we talk about a solution that's hypotonic, it has less solute than the cell, if we're talking about cells. It's hypotonic to the cells. So here, if I have in brown a cell with some material dissolved in it, the extracellular material here, the blue, is hypotonic. It has less solute in it. Hyper means what?
Equal.
So, no?
About.
More. It means there's more solute
Oh wait, I'm not.
than the cell. So here in this far right, we have a hypertonic solution. In that there's much more solute outside than there is inside the cell. It's hypertonic. If you remember from earth science, ISO, when you draw isobars, what do you connect? Equal
Values.
equal values of whatever, pressure or temperature, right? So yeah, iso means equal. So an isotonic solution has the same solute as the cell, equal concentration. So,
Hey, isn't
Isn't
this
this-
mirror?
Yeah, this is
Whoa.
Is that your classroom?
Are you
wait,
promoting your own
So-
YouTube channel?
I set this up. You remember this demonstration?
Yeah,
Oh,
I
yeah.
do. Wait, when was this?
I don't know if this was
This was
Well,
definitely
no,
us.
like-
2 years ago or if it was 3 years ago.
Well, okay, if it
I
was
think
2
it's
years
two.
ago, it was us.
Yeah, so anyway, this demonstration shows a dialysis tubing area surrounded with water, okay, and the dialysis tubing is semi-permeable. So again, we're looking at a demonstration of osmosis because it's water moving through a semi-permeable membrane. Tell me, is this dialysis tubing bad? What type of solution is it in, using the terms we just talked about?
Molasses.
What is it in the graduated cylinder? Pardon?
hypotonic.
It's hypotonic. It has less solute than the molasses. The molasses has a ton of dissolved sugars in it. the pure water around it is hypotonic. Therefore, what is water doing? Going into that dialysis tubing from a higher concentration of water outside to a lower concentration inside, and therefore that forces the molasses up the tube.
Is this in the future? It says 11:18.
Yeah, this is the future. I filmed this tomorrow.
Tomorrow it's 18 and a half days.
In like
from
20 days.
from now.
Oh yeah.
Not tomorrow.
Me and Marty McFly.
Unless he's
Well, no, it couldn't be
investigated.
because a bunch of the pictures are missing from the top.
What picture?
No, because he changed his room up.
All right.
That's definitely
So in a cell, how does osmosis affect cells? Well, it has a profound impact lots of times. Okay. So first off, there are some terms that we need to recognize. Okay. And cytolysis, cytolysis. Cyto means
Cyto.
Like cytoplasm.
cyto. It means cell. Cyto means cell. Lysis, like lysosome. Hydrolytic enzymes.
Digestive.
It means, yeah, basically to rupture, to break down, to burst. So when cells are placed in a hypotonic solution, again, a solution where there's less solute, okay, water, due to osmosis, will travel into the cell. It can lead to the bursting of a cell. And this is an important factor to plant cells. Because what is in the center of most plant cells? Well, no. Nucleus is generally not in the center. There's a large vacuole, right? What goes into that vacuole?
Water.
Water. There's a large vacuole that's filled with water. Okay. And as that central vacuole is filled with water, it exerts pressure, because what's surrounding a plant cell?
A cell wall.
Yeah, the cell wall, which is basically, after it's formed, is it's non-flexible. It's basically a little shell around the plant cell. And so you can imagine as you fill that central vacuole with water, it starts pushing against that cell wall. Okay? And it makes the plant cell what we call turgid. Has turgor pressure. That's pressure of this vacuole pushing against the cell wall, and that keeps plant cells upright. An herbaceous plant, a green plant, and not like a tree with wood and bark, but just a flower, a dandelion that grows out of the ground, something like that, where we have just sort of not woody material, but just a regular stem. Okay, that plant is able to stand upright because of that turgor pressure, because of those central vacuoles are filled with water, they're pushing against the cell wall, and that gives it its structure. And when those central vacuoles lose water, that pressure is reduced, and that's what leads to the wilting of plants. If they don't have enough water, they're no longer able to stand upright because that pressure is gone. And that cell wall prevents these plant cells from bursting, sort of contains that pressure, and absorbs some of it. Animal cells can burst due to this osmotic pressure, okay? And so if we have a red blood cell placed in distilled water, just pure water, water will flow into the red blood cell. Its shape will expand. Its cytoplasm will expand. It will press against the cell membrane. It will lose that disc shape that it normally has, and eventually it can burst, okay? And leak the cell contents in the cell is then basically dead.
Was the E backwards?
What's that? Caleb?
Is the E backwards? Like,
So animals have ways of dealing with this. Body systems have ways of removing excess water. Okay, like the excretory system removes excess water. Single-celled organisms also have structures for dealing with this. An amoeba, a paramecium living in fresh water, has to constantly deal with this issue of osmosis of water coming in, and it does that using the contractile vacuole. The contractile vacuole pumps out, you can see here water coming in, the contractile vacuole swells up and then pumps that water out.
Are we ever going to get to see one of those explode?
No. The opposite of cytolysis is when cells are placed in a hypertonic solution. What happens to cells placed in a hypertonic solution?
They shrink.
Yeah, they shrink, they shrivel because water is leaving the cytoplasm,
More than it's coming in.
okay, more than it's coming in, and the cells lose water, and the cytoplasm shrinks. In plant cells, you have a reduction in turgor pressure, which leads to the wilting of plants. In other types of cells, the cytoplasm shrivels up, the cell becomes smaller. Now, if we look here, we have a diagram showing both animal cell, plant cells, red blood cells, and plant cells. And again, when these cells are placed in a hypertonic solution, they lose water because there's more solute out here, and they shrivel up. They lose their shape, they shrivel up. In a plant cell, again, the cell wall remains in the same shape, but the interior contents of the cell kind of shrink and shrivel up and withdraw from the cell wall. And in isotonic solution, we have just normal, sort of normally shaped red blood cells, a normal plant cell. We have equal movement of water back and forth. And in a hypotonic solution, we have that swelling of the cells. Here, the cell wall contains and withstands that pressure. In red blood cells, you can have the bursting of those cells. All right, we'll stop there for today. I'm not going to get on to cell physiology.